All right, welcome back to part 2 of DNA replication. So where we had left off is we have the origin of replication um, in E. coli. This is E. coli specifically, although most bacteria have a similar paradigm. Um, we have a site that we call the origin of replication. It's also termed ORIC, standing for origin of chromosomal replication. We pointed out that there are 3 different types of sequences in here. One is an AT-rich region. So we have A's and T's rich in these 3 blue sequences. It tends to have more A's and T's than G's and C's. We have several DNA boxes, DNA A boxes. DNA A is a protein that binds DNA, and it is responsible for initiating replication. So here we have one binding site, and here we have a second binding site, third binding site, 4, 5. You'll notice that these all have similar sequences, but they are not identical. Thirdly, we have GATC methylation sites, and these are these underlined regions, and you can see several of them throughout. How this all comes together to initiate replication is we have these regions present at the ORIC. The DNA A proteins will come in and start binding the DNA A boxes. So there's something in DNA A that fits into that major groove, makes contact with the sequences in these DNA boxes, and causes it to bind. As all of those bind in there, they also bind to each other. So DNA A binds to DNA at DNA A boxes, but also binds to other DNA A proteins. This causes the DNA to kind of wrap around this complex. The fact that you have a region immediately adjacent to it that is AT-rich will cause a bubble to open up or a strand separation. The reason why this needs to be AT-rich is, if you can remember or recall, A's and T's bind to each other with 2 hydrogen bonds. G's and C's bind to each other with 3. The more hydrogen bonds you have, the stronger it is. So here, what we essentially have is an area of weakness that is easier for the 2 strands to separate. Once that takes place, that allows 2 single-strand pieces of DNA to start interacting with other proteins. One of those proteins is something called DNA helicase. DNA helicase wraps around the individual strand, and it will help prevent the DNA from coming back together. We also have something else called DNA-C that is binding single-stranded DNA, and it helps hold the process or hold this bubble open. DNA A helicase is also known as DNA B protein. So here we have 2 helicases loaded on, and the helicases use energy from ATP hydrolysis. So we burn ATP, and it causes it to travel in a 5' to 3' direction. The end result of this creates an ever bigger bubble of single-stranded DNA. So here's normal double-stranded DNA, normal double-stranded DNA, so it's double helix here, double helix here. This region right here is single strand here, a single strand here. So it is not in a helix, and we have these DNA helicase molecules moving from 5' to 3'. Where we get these strand separations, we'll refer to that as a fork. And so you can see that we have two replication forks forming. Now, we've discussed what the DNA A boxes do. We discussed what the AT-rich region does. We did not discuss what the GATC methylation sites do. Well, these are involved in regulating replication because we don't want to start one round of replication and immediately start another round. We want it to start and progress on and go through all the fact checking that needs to get done, all the proofreading. So how we do that is we have a protein called DNA adenine methyltransferase. So this transfers a methyl group onto adenine. And it's specifically the adenine that is the A in this GATC. So this protein recognizes this sequence, GATC. It's binding in the major groove, so it can make contact with the base pairs. When it binds, it recognizes the adenine and adds a methyl group onto it. What this does is all of the DNA that was originally there, the parental DNA, has been sitting around with this damn protein, methylating those sites. So when it starts off, all of the DNA is methylated. When you initiate the replication and go through this process and start producing daughter DNA, the daughter DNA is not methylated. It takes a while for this damn methylase to come in and start methylating the daughter strands. So it takes several minutes to even become started. So the initiation of replication only occurs if the DNA is fully methylated. So when you have a new strand being laid down, it is not methylated, and this will block the initiation of this replication fork. All right, so now we've talked about how DNA is initiated at the origin of replication. We've talked a little bit about how it's regulated and the mechanics of how strands get separated. Now we are going to get into the nitty-gritty of how replication actually takes place and the proteins involved. So, learning outcomes for this section is describe how helicase, topoisomerase, and single-strand binding protein are important for the unwinding of the DNA double helix. Again, we are going to have a lot of new words here describing proteins that are involved in specific processes. So here's a few. We'll also outline how primase, DNA polymerase, and DNA ligase are needed to make strands of DNA at the replication fork. We will compare and contrast the synthesis of the leading and lagging strands. And this has to do with the fact that DNA only gets replicated in a 5' to 3' direction. I want you to be able to list the components of the replisome. Replisome is referring to the complex that forms that carries all of this out. And we'll also describe how DNA replication is terminated. We've already talked about how it starts. We'll talk about how it finishes. So let's start off with a few definitions. So DNA helicase, which we've already been introduced to, this is the protein that gets loaded onto a single strand after you get that bubble forming at the origin of replication. So DNA helicase is responsible for moving in a 5' to 3' direction and separating the two DNA strands by breaking the hydrogen bonds that are between them. As you pull the two strands apart, this is going to cause the DNA to unwind, or the helix to unwind. So this generates positive supercoiling ahead of each replication fork. This is going to be a problem in the long term. So we have topoisomerase 2, which we've also called DNA gyrase. This travels ahead of the helicase, so this is in front of the helicase, and it introduces negative supercoils to alleviate the positive supercoiling that would occur. So we're being proactive, DNA gyrase comes in and starts adding extra negative supercoiling to prevent positive supercoiling. Now, these single-stranded piece of DNA, as it gets longer and longer and longer, will want to come back together to rebind. This is where single-strand binding proteins come into play. These bind specifically single-stranded DNA. It does so in a non-sequence specific way. So it's recognizing the backbone, but it binds to it and prevents the DNA from coming back together. Without these proteins present, the DNA would simply readhere with this complementary strand. All right, we will also have a protein called primase, and primase will lay down a short sequence, about 10 to 12 nucleotides of RNA. So the very first step here is to lay down a little bit of RNA that will act as a start site for DNA synthesis. So since it acts as a start position, we'll refer to these as RNA primers. This is something that there are not proteins available to take a naked strand of DNA and just start adding in actual DNA. Only RNA can be laid down initially. And we'll go into detail about that later. So these short RNA primers act as a landing pad for DNA polymerase. They're usually only 10 to 12 nucleotides in length. The leading strand, because of how it's orientated, will only have a single primer. The lagging strand will need many primers in order to carry this out. This concept is a little bit easier understood once we look at the schematic. And then the final player here is DNA polymerase, which is the actual enzyme responsible for synthesizing DNA. It's going to recognize the RNA primer on the DNA that was laid down by primase. So when we come back, we will essentially take all those individual players and talk about how they fit together and how this leads to DNA replication.